let's get started. Okay. So today, it's a great pleasure to introduce Stephanie Huang, who is, the co-leader of a genetic mechanism program of a cancer center. And she is a professor in the School of Pharmacy and, Department of Experimental and Clinical Pharmacology. And she's from Shanghai and got the master and the PhD from Purdue University. And she's been in University of Chicago for some time and came here to Minnesota in 2017. And since then, she's been here. Now she's a tenured professor and she's co-leading the program with me. So today's Stephanie's talk will be interfering therapeutic vulnerability within tumors through integration of a pan-cancer cell line and single cell transcriptomic profiles. Thank you very much. Please take it away.
Well, thank you, Masato, for the introduction. And good morning to all. And it's really nice to see you in person. And I guess it's also appreciated for people attending online. I figure it's been a while that I meet with people in cancer centers. So just to give you the sense of research ongoing in my lab. So everyone who knows my research, I will tell them everything we do is drug related. And, really one of the main theme of the research work ongoing in the lab is to take advantage of existing data. So our goal really, living in this data era, we want to be able to utilize these available information to the fullest possible. So one of the major goals that we have is to utilizing these data and build model around it to allow us most accurately project patient outcome response to medications. And then we apply these projection to large collection of patient cohort information to enable biomarker discovery and drug discovery. So we actually, if some of you might heard some of my previous talk, basically, we're one of the early group to leverage the presence of high throughput drug screening done in cancer cell lines. So there are a lot of effort ongoing. Basically, they are screening large number of compounds in a fairly large, close to 1,000 different cancer cell lines. And all of these tools, the measured drug sensitivity has been deposited in the public domain, so anyone could access these data. In addition, the molecular profiles of these cell lines are also available online. So we take that as a really good resource to allow us to learn the relationship between the molecular feature of cancer along with measured drug sensitivity. We then, building models and using tools to learn these relationship and then apply these relationship matrix towards our patient molecular data that will enable us to project patient likelihood of response to hundreds of different medications. So think about it, in trials, you can do it one drug at a time. You can never test thousands medication in patients. So this kind of approach would allow us to really evaluate drug response in a much larger scale compared to your regular clinical trials. So this type of effort has really enabled us to really amplifying the power of discovery. So, we have a number of successful stories around biomarker discovery. Think about it. TCGA is one of the examples, the cancer genome atlas, where over 10,000 cancer patients has been collected. We know the molecular information, but we don't really know their drug response. By applying our approach, we were able to, for every single one of those 10,000 TCGA sample, we have hundreds of different possible response to medication information. We then can do association study between these molecular markers and the imputed drug sensitivity to allow for biomarker discovery. So we have a number of successful story on that end. We also have applied these for drug discovery. We look for specific subtype of disease that doesn't have good therapeutic options. You can identify drugs that would be beneficial to those subpopulation. You can also look at this in the context of drug resistance. So if standard of care resistant in that setting, what can you do? So these are all different efforts we've taken today, but sort of to change the focus a little bit. All this, things I mentioned before is mostly focused on the patient cohort level, on the population scale. Today, I want to spend the time to introduce you to a new tools that develop in the lab, which we will decide to zoom into single cell level. So in this case, we're not looking at the population and cohort as a whole. We're aiming at projecting drug sensitivity at a single cell level. So why do we want to do that? I don't think I need to convince people in the room that cancer is a heterogeneous disease, right? It's very complex. There's huge inter-patient variability exists, but also when you look into a piece of tumor or cancer specimen, there's high intra-tumor heterogeneity too, both in the context of different type of cells, and when you zoom into the malignant cells, there's also clonal differences, and there's different, genetic mutation in different cell type. So all of this would lead to, we have clear evidence that, in a chunk of tumor, there are some subclones that intrinsically resistant to standard of care treatment. So when you introduce these care, the drug wipe out the population that was responding to it, but then there's residual cancer cell left that will progress and then lead to disease recurrence and drug resistance. So this is a very phenomenon that we all take and acknowledge. But what can we do? Can we identify these subpopulation? I would say in the recent years effort, we are gradually taking on that approach as a community. So single cell RNA sequencing is one of those tools that allow us to have these finer resolution of interrogating cancer. So by looking at RNA transcriptome information on a per cell level, we now can really describe in the chunk bulk tumor, there's different subpopulations. And we can find different genetic mutation. There's single cell DNA sequencing, we can find out mutations specific to a cell. We can also look at this on the transcriptome level. So this really has been, in my opinion, it's really somewhat revolutionized how we can interrogate cancer in a more finer resolution. So this is very cool, and many effort has been done to showcase, either stem cell or certain subtype of cell type would lead to drug resistance or recurrence. I would, like I said, we care about drugs, and we would argue, well, this is really nice that we can identify these problematic subclone or population. What can we do with them? Can we design drug towards those problematic cell groups and therefore was the goal to eventually cure cancer? So that's what where this whole idea about designing these tools to be able to project single cell level drug sensitivity come from. So, like I said, our goal really, we want to translate these single cell RNA-seq data into projection of cellular susceptibility to therapeutics. How do we go about doing that? Again, similar to the patient cohort level, we can't. Right now, the gold standard is that people are spending a lot of time and effort and money into tracking these single cell and looking for their drug sensitivity. So there's, people barcode, either you get a tumor sample, you can separate them by single cell profiling, and then you can expose different single cell population to different drug and measure the drug sensitivity. So this is, I think, the field recognized as a gold standard. But imagine any of you who has done single cell sequencing data, how expensive it is, and not only the price, but also the effort, time involved to make sense of those data, and then eventually also, with drug treatment, that's another layer of effort to determine which, and you can only do that as of now, one drug at a time. So looking at the field of literature, I believe we've done a very careful effort in trying to exhaustively identify these measured single cell level drug sensitivity information. Maybe we get a handful, no more than 20 of these data sets. That's currently in the literature. You think about it, we have hundreds of different medication at hand. We want to be able to utilize all these possible tools and likely new drug coming on board too. So we thought, well, in the absence of measured single cell sensitivity towards drug information, can we use computational effort to project the likelihood of response at a cell level, which will really provide a lot of information for us to design drug treatment. So in this case, again, we resorted back to the cancer cell line tool where, hundreds of drugs has been exposed to these cell lines. We have the molecular data there. So we already know through previous cohort level work, we can build relationship between transcriptome and measure drug sensitivity. Well, can we use that relationship towards single cell RNA-seq data, then projecting the likelihood of response at a single cell level? So that was the idea here. But it's not, the initial thought is pretty straightforward. However, to really execute it, it's, there's a level of complexity. But before I get there, I want to introduce the type of data we have. So I mentioned, bless you. We want to leverage the cancer cell line drug screen. So for this particular study, I want to introduce you to two different type of cancer cell line drug screen. Those are the first bullet point is of the Broad Institute, U.S. Broad Institute effort, so CCLE and CTRP. So CCLE is a cancer cell line encyclopedia. So in this data sets, they have collected over 1,000 all cancer cell lines. They have done molecular profiling on these cell lines. So we know the RNA-seq information for these 1,000 cell lines. We have genomic information. We have actually more recently proteomic information for these cell lines. So there are a lot of rich molecular data available in the CCLE. And CTRP is an extension of CCLE where, so they basically utilizing this collection of cancer cell line and expose them to drugs. And when I say exposed to drug, it's a gradient. So they do a increasing concentration of drugs and measure the IC50 growth inhibition in each cell line. The reported metrics including IC50, which is the concentration that required to inhibit 50% of the cell growth, as well as AUC, that's the area under the dose response curve. So together, CCLE and CTRP has presented over measured drug sensitivity for over 500 drugs, and also molecular information for close to 1,000 cancer cell lines. And in parallel to the broad effort, the Wellcome Trust Sanger Institute in the UK has also started a GDSC, genomic drug sensitivity. So it's very similar effort and done at the UK. They, again, getting this large collection of cancer cell line, have done molecular profiling, and they have measured drug sensitivity in these in these cell lines. And I would treat these two efforts sort of, oftentimes in my lab, we use one as a discovery, the other, the drug doesn't always overlap. So you see 500 here, 300 here, maybe about 200 are overlap. So if you want to expand it, the pool of drugs that you're interested, you can do both. I didn't list here, but another thing that we've been used is the recent PRISM, which is another broad effort. They have had much larger pool of drug screen. So PRISM has screened over 4,000 drugs. And the nice thing about PRISM is that, so both the two listed here are mostly focused on anti-cancer agent. The PRISM screen has expanded to non-cancer, non-oncology drugs. So of the 4,000 drugs, many of which are non-oncology drugs that has been screened. But I would say, from the data point of view, these two data sets are much cleaner than the PRISM screen, because that one was much, their goal was high throughput. They pull cell cell line together to screen compounds. The measured noise level are higher. So there's a give and take, depends on what you're interested. You can choose. There are a number of these high throughput drug sensitivity cancer cell line screen that's available, so you can take advantage. So that's the sort of the end that we can learn about measured drug sensitivity and molecular features. Regarding the single cell RNA-seq, as I mentioned, we have tried to look for these what you can call the gold standard type of measurement. So the first three I'm listing 6 different data sets here. We have a few other, but the idea here, we want to use data that come from a variety of different sources representing different type of cancer, maybe different model system. As you can see from this, the top 3 are cell line basing, and then we also have organoid culture as well as cell cultured in different microenvironment and PDX and sort. So the first 3, the number 1 is a lung cancer. They use a PC9 cell lines, and you can see from the drug column, there are a number of different drugs that has been assessed, some targeting agents, some cytotoxic chemotherapy. So what many of these data sets, how did they set up? They have a cell line that's in, so the parent cell lines are sensitive to the treatment. So then they chronically expose these parent cell line with the drug of interest. Over time, the cell line become resistant to that medication. So then they do the RNA sequencing, single cell RNA sequencing, both on the parent cell line as well as the resistant cell lines. So then you have the knowledge was each population are distinct in their sensitivity towards the drug of interest, and their single cell RNA-seq profiles are different. Therefore, they're looking for what make them different, and they use these data set to look for reasons for resistance. So the first 3 are lung cancer, AML, and also breast cancer. We actually use these 3 data sets as our training data. And then the last 3, one is in PDX model, one is a cell line, one is testing microenvironment, which I will share with you a little more, but those 3, we use those as a validation data sets. Yeah, so once again, let's get back to our method of interest. So on the top, these are the high throughput cancer cell line screen where we have the measured drug sensitivity. This is a representation of one drug exposure in one cell line. As you can see, with increasing concentration of drug, the cell start dying. So the growth inhibition curve is showing here. And in this particular study, we use the area under this curve as an indicator of drug response. As you can imagine, the higher the AUC, the area under the curve, the cells are more resistant to this treatment. If you have a much smaller area under the curve, that suggests very little drug were needed to inhibit the growth of the cells. So high AUC meaning more resistant, low AUC meaning more sensitive. And then we have the transcriptome information measured through bulk RNA-seq in these cell lines. And we also have the single cell RNA-seq data. Right away, intuitively, well, can we just build a model and apply that to the single cell? When I say it's complicated, the answer is, so for those of you have done bulk RNA-seq, so the bulk RNA-seq basically is taking an average expression level for everything in your sample, right? So it's not specific for any cell type or any location of the cell. It basically taking an average of what you get from whatever chunk that you're evaluating. On the other hand, single cell, the whole beauty about single cell, we were able to capture the spatial, the location, the temporal. We are also capturing these finite distinction between cells. So that's the beauty of single cell. Therefore, by directly utilizing bulk RNA-seq data towards single cell, you're losing all these information. So that's one of the first hurdle in this sort of transformation. Second thing, for anyone who has done single cell RNA-seq before, there's, in general, the expression level on the per cell level is way lower than what you can get from the bulk signal. And also there's the dropout rate for any gene. Sometimes it's real biology. It's the cell type that missing that gene. Other time it's just because the detection limit and other variety of reasons will lead to a high dropout rate for genes. So in the field, there are thoughts about how do we integrate bulk RNA-seq and with single-cell RNA-seq data. There are two schools of thoughts. One, especially related to drug sensitivity, people has try to use the bulk data to generate signatures. So if in the bulk data, you have out of the 20,000 genes, you have these 100 genes that was associated with this particular drug sensitivity, then apply these 100 gene signature to the single cells and then allow projection of the likelihood of response in the single cell. This is one type of thoughts towards, transferring the bulk to the single. I would argue, because that non-random dropout rate in the single cells, so out of your 100 gene signature, maybe 80 of them are not there. Can you say that's because, well, if we can confidently say these 80 gene doesn't express in the single cell, that's perfect, that we achieve our goal, but that's not, nobody can confidently say that. There's all these random dropout. So that lead to the performance of along that line of work, not very reliable. So because of that non-randomness of dropout in single cell sequencing. The other school of thought is to first try to integrate using deep learning or neural network type of thing to first integrate bulk cell RNA-seq with single cell sequencing. So that will allow you to learn something in common and then apply that towards drug sensitivity screen, which this has been really useful, but the problem has been, when you think about deep learning, you want a lot of data. So in this case, sometimes single cell, when you look at, if you look at the previous table, the scale of number of cells we have is ranging from 500 to 5,000, sometimes even more. We have millions of cells when the study were done in the more, you can argue it's higher quality or what. But when we think about cell lines, and we're still dealing with maximum of 1,000, it might sound like a lot in our regular day-to-day. When you apply deep learning, 1,000, it really isn't much at all to be able to train your model to come up with those robust parameter to interrogate the truth. So with those different thoughts, we decided, well, why don't we do it in the way that we want to integrate bulk RNA-seq and single-cell RNA-seq. We also want to select gene that matters because, we all would agree not every single gene in the genome would lead to relevant drug sensitivity, right? So if we could somewhat filter out the more informative gene list, we will have more information. So instead of taking everything into the pictures, which we need a lot to train the data. So this is what we come up with. We actually nickname our model SciDuck. So I will refer to SciDuck stand for single cell integration and drug response calculation. It's a lot to say. So, and my postdoc, my student loved the Pokemon SciDuck. so we've been nicknamed this tool SciDuck. From now on, I will refer to that. So what we did here really, first, we want to come up with an informative gene list. So this is all done in the bulk the cancer cell line tool. So we have measures drug sensitivity, we have gene RNA-seq data in these cell lines. We are doing, using Lima, we're selecting genes that could contribute to measure drug sensitivity. But in this case, one tricky thing is some of the previous model, when they select signature, they set a p-value cutoff. So they say, we'll use p less than 0.5 or whatever. This is the number of gene. I bet you nobody can translate that p-value to clinical usefulness, right? So we decide not to do that. This whole exercise is give us a rank order of genes. So we're not setting the cutoff, whereas just this is to provide information for every single drug. What is the degree of a given gene that were related to that drug? This is the piece of information we're collecting. We'll use it later on. And the second step, well, in this case, we call first step, but this is a single cell RNA-seq, and this is a bulk RNA-seq from cancer cell lines. We do these integration. And then after integration, what we are aiming to get is what is in common between the single cell RNA-seq and the bulk RNA-seq. So we are converting these RNA-seq data into two components. One was the shared component. The other is what's unique in the single cells or unique in the bulk cells. So then the data was, instead of one chunk data, we're transferring them into the shared component and the unique component in the single cell or in the bulk space. And then we use that transferred RNA-seq data as our input to later on train how they related to pharmacological trait. So to do that, we also have, so using these, so basically the machine learning to, there are different tools, and I'm listing here the NMF and CCA. Those are two different approach to allow for data integration. We then to train the information about drug-related gene lists will come in here. So there, what we test the parameter is the ratio between number of single cells and the number of genes. So instead of choosing a p-value, calling these genes are drug-related, we think about it. Some single cell database has 500 cells, other have 5,000 cells. So if you apply the same 100 gene into this projection, there's different degree of accuracy, right? When you have more sample, because we all know, well, even if you haven't taken the statistic, right? If you have 20 sample, you have 20 parameter to predict. That's the maximum you can get, right? Like you want to have less than your number of sample of predictors to do this calculation. So that's where the gene expression information. So we're taking a ratio between the number of sample and number of informative gene lists. So we tested in what range can we do this right. So there are two parameters we're testing in the building of the model. Which model is better, giving us better integration between bulk and single cells, and also what is the number of single cell to gene ratio that's more informative to allow us to accurately project a drug sensitivity in the single cells. So let's look at the performance. So a heads up, today I don't have a tons of slides, but every single slide requires some time to explain. So bear with me. I want to spend time on each one of them so you can understand what it is. So this is basically evaluating the performance of our model. We are doing this in 3 different models. So the top one is the lung cancer PC9 cell line screen for gefitinib, I believe. The second one is the sorry, I can't even see it. Oh, this is a breast cancer MCF7 cell line exposed to, I believe, bortezomib. It's a proteasome inhibitor. And then the last 2 samples are AML sample exposed to an IBAT inhibitor. So both. Just to recap, in all 3 data sets, they have chronically exposed these cancer cell line with the drug to develop sensitive and resistant clone, and then they do the RNA-seq, and they know a single cell RNA-seq. So they know which population is sensitive, which population is resistant. What we did here, we just take all these single cell RNA-seq data without knowing whether they are from the sensitive group or the resistant group. We apply our tool to this whole collection of single cell sequencing data and ask the question, how many times do we get it right when we call a cell sensitive to that drug when it actually is determined to be sensitive? And then so we did the panel on the, I guess on your right is the one, we use 2 different common language effect size measurement. In this case, the row statistic and also the Cohen's D's effect size. So this is, don't get bugged on these things. Think about it like a t-test, like the row statistic is similar to a p-value. The Cohen's D is kind of like an effect size, like the beta you can detect. So the bigger they are, or the more significant they are. So we're looking for high value on these charts for all of them. So what we're trying to say is if by applying our model to a single cell RNA-seq data pool, if we can tell apart the sensitive cells from resistant cells, and that's matching to the gold standard expectation, then we got it right. And so the more significant we are is to say we can separate the sensitive from resistant very clearly. So we are looking for a high value on both the rho statistics, think about it as p-value, and the Cohen's d, that's the effect size. So just I apologize, these are small letters. So the rho statistic is actually ranging from minus, so it's from 0 to 1. So if you get a 0.5, that's a random guess, right? You could half of the time you get it right. So you're looking for values above 0.5. And the Cohen's d is you're looking for differences between sensitive and resistant thing. So the bigger the difference, the more likely you're separating the sensitive from resistance. So in this case, for every single graph, I'm showing 3 different colors and representing 3 different methods. The gold grids are those ones that without any integration. Basically, what we did there is to take the relationship learned in bulk RNA-seq and measure drug sensitivity, apply that directly to single cell. If there's missing data, that got dropped out. So this is without any data integration. And the maroon color and the blue color was our method, but the choice are one of the 2 different data integration methods is either NMF or CCA. So we were curious about these different transfer learning tool, which one would give us better result. And then on the x-axis, we are evaluating that ratio between the number of single cell to the number of genes that we have. So again, you can think was closer to the y-axis, this is a higher ratio, and then the very low, like about 0.2 is on this end. So that is to indicate we have way more gene than we have in cells, versus on this end, we have more cells than the gene, informative gene that we have. So as you can see, I almost expected, when you think about just on the x-axis, if you have more predictor, in general, you would do better, right? So we are seeing higher value towards this end of axis compared to this end. And then on the y, and right away, you can see this big separation between the gold bars to those blue and maroon color. So number one thing takeaway from this chart is that without integration, you're doing barely just like a guess out of what the likelihood of response could be. So in data integration between bulk and single cells are very, very important. That's the case for all 3 data sets. Number 2, we actually come up, and then we asked specifically which method is better. For the lung cancer data set, they're very comparable. You can't tell apart, but for example, the IBET, wait, which one? One of so for the most part, the CCA method is outperformed the NMF method. So we decide to go with that integration approach, and we also choose these ratios sort of towards this end to yes. Oh, I think so. I had the laser, but now I lose it. I don't. Yeah. I don't think I had just for those of you online, I'm trying to point at things, but I think my screen is frozen. Anyway, so this one is very, very technical, but I think if without knowing, I want to spend time explaining it, but the take home point really is when you want to leverage bulk RNA-seq data into single cell space, some form of integration is very critical and very important. And number 2, there's determination about, the sample to predictor ratio is also an important factor to take into to affect your model performance. Oh, wait. My PowerPoint is freeze. I'm gonna restart this. Let's see. Okay. I restarted my PowerPoint. Let's see. Oh, it worked, I think.
Are we on Zoom?
People on Zoom, can you see I'm advancing the slide? Okay, thank you. So we just went through this very complex graph. Again, just want to say the take home point is about data integration is necessary, and we need a really good ratio between the number of sample and number of informative genes. So the next question is, now we have a method, we can tell the sensitive from resistant cell line apart. How is our method fair to other existing methods? What I can tell you is there are really very, very few existing methods that's out there to do this type of thing. So I'm listing two methods here. One is this turquoise color called Beyond Cell, and the other one's a Cataracts. That's a so Beyond Cell is I think it's a signature base, but what it did is they use a lot of training looking for the most accurate signature for a given drugs. So they basically use single cell drugs screen data to train the model, so they can come up with a collection of signature that's very accurate towards that particular drug. And then they use this to project the data for the, well, actually both Beyond Cell and Cas, none of those previous methods have applied their method outside where they developed the method. So this is one of the difference there. But Beyond Cell, basically there's a signature-based thing versus Cataract, it's signature and also doing some data integration. So the blue and pinkish one are our methods, the SciDux method, and we chose different criteria within SciDux just to see how robust is our method, you know, if we tune this different parameter. As you can see on the left, sorry, your right is again, the row statistic representing p-value, and on the left side, it's the Cohen's d, that's the effect size. Once again, we're looking for higher bar is the better performance. And you can, you know, right away see that our method, the SciDux method, is outperform the other two methods across the board for, in this case, we apply it to 3 independent data sets. So none of these are training data set. We did this for training data set. We see our method, of course, outperform the other method. We also apply these to independent data sets. And in this case, again, our method can outperform the other 2 existing methods. So, oh gosh, it freeze again. I don't know why my screen is not moving.
No, it's just.
Okay.
Yeah.
Okay. Maybe fall asleep. So now we have a method, and we show that it does better than some existing tools. The key is to apply this thing. So I am going to give you 3 examples how we apply SciDux towards clinical questions. The first major thing we care about, you know, thinking about cancer intracellular heterogeneity, you know, we mentioned there's an intrinsic drug resistance. So the hypothesis, there's subpopulation presence in the bulk tumor that's intrinsically resistant to standard of care treatment. So in this case, we are curious, can we find drugs that could be efficacious targeting those subpopulation that will be intrinsically resistant to standard of care? So that's the first example, the SN38 resistant in the rhabdomyosarcoma. This is a pediatric condition. It's rare, and it's been treated. Standard care treatment is irinotecan, and the SN38 is an active metabolite of irinotecan. I will show you what happened there. The second example is docetaxel resistance, castration-resistant prostate cancer. So this docetaxel is a standard of care for castration-resistant prostate cancer. In the second example, I'm showing you the acquired resistance. So in this case, it's not the first exposure of the drug that lead to resistance. is the chronic exposure to the standard of care that lead to resistance in the tumor. So again, in this case, we're looking for once you develop resistance to standard of care, what agent can you use to treat? The third example, I'm actually showing you an evidence that the CyDex model not only can capture, it can capture cellular response to medication. It's not just the molecular structure of the malignant cells. It's also because transcriptome is, I mean, I think that's one reason the lab decided to focus on transcriptome. Transcriptome reflected the DNA molecular landscape, but it's in response to surrounding environment. So in this case, I will give you an example. CyDex method can capture the same cancer cells, different drug response after exposing to different microenvironment. So let's get to it. This is the very first example. So RMS is like I said, is a pediatric malignancy. Our collaborator at the St. Jude's Children's Hospital has been collecting these patient samples, and they have done single cell RNA-seq data on these patients. They found actually in their work published in last year in developmental cells, they found there's a subpopulation, the mesoderm a subpopulation of these tumor, they are intrinsically resistant to standard care treatment. So the majority of the bulk tumor, that's the myoblast cells, they are sensitive to irinotecan treatment. But the subsets of mesoderm cells in the tumor are resistant to irinotecan treatment. So they posed the question to us, can you find drug from your pool that would be efficacious in these resistant population? So what we did, as I mentioned, we have different large scale high throughput drug screen. So we utilize both of them to independently to generate our drug prediction. What we did is we take once again, all the single cell data from those tumor. We did it one patient at a time. They have 11 patient total. So say in one patient, we generated predicted sensitivity for every single line in every single cells in that patient. And then we separate that cell, the patient tumor into mesoderm cell, which is the one resistant to standard of care, and the myoblast, that's the sensitive group. And we asked the question, is there any, and we did this for every single drug we have, and we asked the question, is there drugs that showing higher sensitivity, which meaning low AUC in these mesoderm cells compared to the myoblast cells? So in this case, and that's the discovery I'm showing you here, when we do this in the broad data set CTRP, we found 7 of the EGFR inhibitors show up to be potentially sensitive in the mesoderm cells. And also there's mTOR inhibitor and BRAF inhibitor. And when we did this in GDSC data set, once again, you see EGFR inhibitors show up. And very cool thing is that our collaborator, when they publish their work, they randomly throw in a few drugs that in their field they're potentially interested. One of those, one class of those are the EGFR inhibitor. Out of everything they tested, EGFR inhibitor is the only one showing activity. So we have on that level a proof of concept that we computationally projected EGFR inhibitor could be efficacious in treating these standard of care resistant cell population, and that has been validated by our collaborator. The table just showing the overlap between these two data sets. We take the top 5. One thing I need to mention, we did this one patient at a time. We're asking for the drug that has to show up at least in more than 50% of the patients. So in that scale, we're finding things that's not only the intratumoral, but also in the cohort level has to be efficacious. So that's the story for intrinsic resistance. This is the one that we did. So the initial single cell data sets, they exposed PC3 or DU145, it was docetaxel, and then chronically developed resistant. These are prostate cancer lines. And so they're doing RNA-seq in both the sensitive and resistant population. So what we did is, again, we take all their RNA-seq data, generate prediction, and we're looking for drugs that will show higher sensitivity in the docetaxel resistant subgroup of these cells. And this is sort of a visual example. So the y-axis are the drug of interest AUC. The x-axis is the sensitive or resistant status for docetaxel. So in this case, if they are resistant to docetaxel, we're looking for lower AUC of our drug of interest, which suggesting higher sensitivity in this group. As you can see here, I'm showing you vemurafenib. It's a BRAF inhibitor, and this BRAF inhibitor is showing higher sensitivity in docetaxel sensitive, resistant group. So once again, we did this in both CTR2, both high throughput drugs, cancer cell line screen. Not surprised, once again, we see BRAF inhibitors show up as our top choice. Both cases, we have 2 of our top 10 drugs being BRAF inhibitor in both data sets. We then went into the lab to say, can we confirm this phenomenon through experimental testing? So in the lab, we created DU145. So that's another prostate cancer cell lines. We did exactly chronic exposure to docetaxel and make the DU145 resistant docetaxel. And then we treated both the parent line and the resistant line with vemurafenib, the BRAF inhibitor, and you can see there's a nice separation of the curve with p-value less than 10 to the minus 4, and there's almost doubled IC50 value for the docetaxel sensitive line when compared to the resistant line, which is more sensitive to vemurafenib. This is the last example, but personally, I found it really, really exciting. So this is, in a, again, different disease setting, pancreatic adenocarcinoma. So what the original study they published in Cell, what they did is they have a hypothesis. The same tumor, when you expose it to different microenvironment, they will show different response to medication, which to me is a very cool, it's reasonable and very cool idea, and they have experimental data to support that. So they expose their pancreatic cells to either sits in the basal media or the classical media. So they show in these 2 different media that either the cell or organoid will display different characteristics. And they also, on top of that, they expose so they have done a whole slew of single cell RNA-seq with these cells to say, well, after exposing to different culture media, do you change the transcriptome of this exact same cancer cells? And then on top of that, they treated the drug in the presence of either classical media or basal media with a collection of drugs. So what I'm showing you on the right, on the left, your left, is the collection of drugs that they tested in their setting. So on top, if a drug going up, that's meaning they are more resistant in the basal media. If they're going down, they're more resistant in the classic media. So you can, and the arrangement of drug depends on the degree of, so not only is a yes and no answer, it's a degree of how resistant. So the higher they are, they're more resistant to this particular drug in that media. So you can see they've shown gemcitabine, paclitaxel, SN38, and then also some other drugs are all, so the top 3 are clearly more resistant in the basal media. And then the MK1775 and 5FU are somewhat moderate, more sensitive, more resistant in the basal media. On the other hand, afatinib and trametinib has shown more sensitivity in basal, but more resistant in the classical media. So we basically take the single cell RNA-seq data that they generate for the same cell, either in the classical media or in the basal media. And we ask the question, can we find the same phenomenon in this case? I was quite nervous doing this because I don't know the level of sophistication we can capture with our method. But the good news is for the most part, we recapitulate the experimental finding from that cell reports. You can see antisense 38, paclitaxel, and gemcitabine show up to be quite resistant to in the basal media based on their single-cell RNA-seq data. And then similarly for afatinib and trametinib, we see more resistant in the classical media. This to me, it's really huge because when you think about single-cell RNA-seq for drug exposure, it's expensive, it's time-consuming. You can still do it, all right? If you want to do this in the microenvironment, there's so many perturbations you can do, and so it's not just a added up effect. It's 10 times, 100 times more effort in order to get these one condition modified in the tumor microenvironment, and that this one study, I think it cost them an arm and leg to do. But if we really have a computational method, anything you can give me one condition, and as long as you have that RNA-seq, I can tell you the likelihood in that condition for hundreds of different medication, what their likely reaction in that microenvironment. I think that really is a very efficient way to narrow. I mean, I still think experiment is necessary, but it's a really quick way to narrow down what condition really matters, because there's so many things we can test. There culture media, cell-cell interaction, there's other, variety of different things. So I believe these computational methods can really help us narrow down to from a collection of possible hypotheses to one that's more likely happen, and for whichever drug that might maybe matter in that case, then allow really speeding up these study of interaction between tumor microenvironment and drug response. So I think together, I hope I showcase this new method. It's called CyDuck. It's able to accurately projecting drug response at a single cell level. Our goal right now, we're applying this tool to a number of large collection. We're doing this in human tumor atlas, so not the H10 data. So that's the newest effort from NCI. They're actually doing so passed from TCGA. Now TCGA is on the population level. H10 is really zoom into the fine resolution of cells. So they pick a few cancer type. They're doing single cell sequencing on these cells, and they're looking for the progression of disease. They're collecting much better molecular and treatment information in these patients. So, so we're definitely putting our effort into H10. We're hoping to overlay these potential pharmacologic information onto another newest NIH initiative to collect molecular data. We want to lay these pharmacologic information on that. We also start to explore this in GTEx, which is normal tissue, because in drug development, we keep getting asked about side effects. So we're at the point we can't ignore that. So we start applying our computational tools to a whole bunch of normal tissue. We want to project the likelihood of response of medication in a variety of other tissue types. So that way we can also, beyond the anti-cancer efficacy, we can also provide information on, normal tissue impact of these things. So our method, again, back to our method, it has been outperforming other methods, even though there are only less than a handful of things that's out there. And we're the first to apply these tools towards clinical question solving. I hope I show you we can really utilize this for a number of things, a number of clinical applications. So we're very open for collaboration or team up with any of you for, looking for interesting question that we can solve together. So that's really, again, it's a team effort. This is really my talented grad student and postdoc and people in the lab. They're the creative driver of things, and to make all these things happen. My big thank you to the team and to our collaborators and founder. Thank you.
I've got microphones.
Yes, Scott has it.
I was going to say Scott can go first because I usually take too long, but I'll go ahead. So yes, thank you. That was really phenomenal, and I'm glad I sort of got to understand it a little bit better. And I think that in my mind, I can think of many data sets that are actually taken from like patient samples where we have like pre and post treatment, and we know, which patients responded and which didn't at the single cell level. And so at least in my field, it feels like this is really like ready to be, applied in a really real way to say like, what drugs should we have added to standard of care? You know what I mean? Based on these predictions, and we could test them. So that would be really exciting. And it seems to me, I guess I'm trying to think about what work, or do you need to do any more work to do that? And it seems like the data integration between the bulk and single cell was like a really critical step.
Mhm.
And it was really critical to get that right.
Mhm.
It seems,
Yep.
right? Because some of the other integration methods didn't work very well.
Right.
But the integration method that you used applied sort of cell line or bulk data onto like some very well controlled, like experimental, mostly in vitro and some like in mouse data. And I'm wondering, do you expect that those integration methods should translate like sort of seamlessly into like patient samples, or do you think that you will need to work out that level of it to actually apply it to patient samples?
Very good question. I think technically to apply this to patient single cell data, we can do this right away. I don't see any technical barrier to do this. How true, within the lab, we have this debate. What we say for sure is to compare to those gold standard we're comparing to. So far, all these, what I can confidently say is that when we look for these data set has measured drug sensitivity, when we apply our tool towards it, we can capture them all. So all the things we have had it in hands, but that is probably less than 10 different drugs because people hasn't just, with single cell sequencing has been done quite, we can get a lot of data there. However, single cell with known drug sensitivity, like, what I mean is not just the patient responded to medication or not, but rather we know this group of single cells are indeed sensitive or resistant to medication. That level of gold standard data is not there are only limited amount of that data. So when we apply, what we can say confidently, when apply our method to those gold standard, we can capture them all. There's still residual question if I move beyond those 10 drugs, because right now we're not predicting 10 drugs, we're predicting 500 drugs. How true is that for the drug that wasn't compared to the gold standard? I can't say was 100% confident. I think it makes sense if we can, because the 10 drug we have, there's cytotoxic agents, there's targeting agents, there weren't just one drugs, and we can capture it all. So that's where we stand now. So I think on the application end, we can do that right away. We have to be cautious in interpreting how true that is. So we're doing this with our Senju collaborator because we're projecting those MACI beyond the EGFR that they have evidence. We're projecting MACI to be the new drug that could overcome. So we're doing additional experiments. So for that, we don't have gold standard to compare to. So we're going back to the lab and doing the mouse, and we're treating it with standard care and MACI to see, can we cure both population?
I guess I'm thinking about, like in AML, for example, we have single cell RNA-seq data at diagnosis, and we have it at like day 30, and we know which ones of those achieved remission and which didn't. And so I'm thinking if you apply your method, right, and you predicted percent of drug resistant cells at diagnosis, you would have a real gold standard answer, right?
Yeah, I think so. We can definitely do it. And I would expect to see, there's change in the standard of care response in that,
Well, I guess just like a proof of principle, like, is your method working in AML, for example, in real patient samples, right? So you apply it at diagnosis, you predict the percentage of drug resistant cells, what drugs they get. It's all the same drugs.
Yep.
They all got the same drugs. What percentage of them are resistant at diagnosis? And then the answer.
Yeah.
You know what I mean?
So, yeah. And we could also test it with your, whoever relapse, like, don't tell me who relapsed. And then we can say, well, this person is likely relapsed because we see more resistant to the standard of care. Let's see whether that's true.
Right. And so like those data have been published, you know what I mean? You could do that.
Yeah, cool. Thank you.
Thank you.
Kind of following along this line, does the SciDuck method kind of require a clonal evolution model where at resistance, those cells are detectable at baseline pre-treatment? Does it account for a scenario where the drug may change the gene expression program in some or all cells, a plasticity type of a model, where that program didn't exist at the beginning, that program was induced by the drug treatment?
I have to think it harder. And the surface level, I don't think we can capture that. So we capture whatever is in that single cell sequencing data. So if your primary, the initial sample has very limited or almost non-presence of whatever features in there, I don't think we can, it's not a capturing of what the drug did to the tumor. Rather, at that snapshot of time, this transcriptome profiling reflecting the status of that tumor. So my guess would be we can capture, if you have multiple sample, we should be able to start showing that, but I don't know with just one sample at a stage, we will be able to capture that. Yeah.
It's definitely a great talk. Just quick kind of follow up too is when you say, for example, one of the SciDuck that you see like BRAF inhibitor sensitivity, have you gone back to the data and see what is the cell population that you're actually targeting in terms of, I'm trying to get an understand for how many cells are you actually killing in the population, and what is the existing kind of population that is actually sensitive to BRAF inhibitor, for example?
Yeah, that's an excellent question. I think that's exactly also, the collaboration direction we're looking for. This straight answer, we didn't do that because that's a published work. We don't have access to those, we basically just take published RNA-seq, single cell RNA-seq data. But that's not to say, similar to what Zohar just asked, like if we can find in these AML population that, this number of patients tend to be resistant. We see them tend to be resistant to standard of care. We projected, maybe a second drug would work for that. And then we can go back because we can, with single cell RNA-seq, we can then look for the cell population. Visually, what I want you to picture is, you have those different color, maybe, red color is one subtype, when blue color is the other subtype. But we can also do this on the drug sensitivity. So we want to say the red color one is more sensitive to, and then between red or blue, whatever, there's going to be shared molecular feature. There will be something different. We then can interrogate these two population molecularly to say what's the difference. But that really requires a team effort, and we're definitely open to do that. And that definitely suggests a direction that we can move forward with.
Yeah, I have two questions. I'm working in the field of pancreatic cancer, which is a really stroma-rich tumor. And then I wonder if this method is strongly effective in case the indirect effect via the intervention to the stroma kicks in. And another question is when you think about analyzing the solid tumor patients' samples, one of the issues we have is because the tissue is small, and particularly if we go into the single cell seq, we can increase the sequencing depth, but there we have a repetitive sequencing, come up again and again. Not can go dive into the low express thing, but significant change kind of genes. So how can we deal with this?
Okay, so the first question about the stromal involvement, I really see it sort of in parallel to that example I gave. That one, they're doing different culture media. We actually, through an NCI pilot project, we are one member to look into it because of our method. So what we are founded to do is looking at these spatial or microenvironment interactions. So what the idea is, I can see stromal, sort of you swap out the culture media, that's the small. So we are generating hypothesis. If you have a experimental condition with different number of stromal cell exposure to your cancer cells, we will do RNA-seq and then we'll generate our prediction. We then go back to that to test, is our projection true? So we are in the process of doing that. Again, I think it's really cool. We have preliminary data, again, in lung cancer, we've shown I didn't have time to show this. So basically we look at the spatial transcriptomic sample. We can actually depict the drug sensitivity at a spatial thing because they, again, have the gold, our collaborator has the gold standard. They know drug on the surface of the tumors are more sensitive to the agent and in the core is not. Based on their RNA-seq at this location, we can also showcase. So I'm fairly confident our tool will be able to accommodate sort of that's exactly the direction I think will be really useful to look. The second question, I'm not fully, can you help me?
Yeah, particularly the single cell seq over small samples, particularly, some low expressing genes, which has a significant role for cancer or, the response to the drugs, we cannot get enough data.
I don't have a perfect solution to it. It is true. I mean, in our line of work, the more cells you get, of course, you can do better in prediction. You capture more. I don't know what the limit of our method is. We can try. Again, it has to be set it up right. Right now, I don't know what the minimal number of cells we'll need to confidently say this will work.
So any other question from online? No.
Okay.
Let's close it down.
All right.
Okay,
It's time.
it's about
Thank
time.
you.
Yep. Thank you very much, everyone.